Good morning, everyone. Great. Let's get started. So we have exciting talks lined up for this morning on the topic of reward and reinforcement learning. So we have two microphones set up, one over there, the other one over there. So if you have any questions, you can just come to the microphone for questions. All right. So let's actually get started. The first talk of today will be given by an invited speaker, Alex Pouget, titled Neural Compositionality at Last.
All right. Is my mic working? Yes, good. All right. Well, thank you so much for having me, and thank you to all of you for being here at this ungodly early hour. I've learned over the years that when you give the first talk at the conference, it's actually really good, because there are only two kind of people who come at 9 a.m. The people who want to hear your talk, and the people who have been partying all night on amphetamines and can't sleep. Either way, it's great because they're super attentive. So thanks for being here. So I'm going to be talking indeed about compositionality. And I want to warn you from the get-go that this is a perspective talk. And this talk is the result of many discussions I had over the years with quite a few people, but in particular the current members of my lab. So while I'm giving the talk, this is very much a team effort. So let me cut right to the chase and tell you what the main message is going to be. I'm going to start by reviewing some arguments that have been offered over the last 30 years, suggesting that higher cognition, which some people nowadays might call general intelligence, is fundamentally compositional. So as such, compositionality should be a major topic of research to a conference like this one. We're here because we want to understand the neural basis of intelligence. If you look at the word cloud that you saw yesterday from Bing's introduction, there is no compositionality in there. It doesn't appear anywhere. And indeed, there's no more than like a handful of abstracts that are dealing with this topic at the conference. So why is that? And the reason is actually quite simple. It's up until about 5 years ago, we didn't really have computational tools to model the neural basis of compositionality. And the main message of this talk is that times have changed. We're at a critical time, if you ask me, in neuroscience, because we finally can start looking into the neural basis of compositionality. And my main goal today is to convince at least some of you to start thinking about this, develop new models of compositionality, design new tasks to look into the neural basis of this fundamental principle. Now, of course, none of that makes any sense unless you already know what compositionality is about. So let me try to illustrate this concept with a very simple example, how to add two numbers. And this is an example that I'm borrowing straight from books and articles by Randy Gallistel, who has been trying to push his perspective for many years. So I don't think I need to remind you of how we add two numbers. We line them up on the right here, and then we take the first two digits, we add them together, get the first digit of our answer, we move to the next column, repeat the operation. If we have a carryover, we take it to the next one, and we keep going that way on the left-hand side. Okay, so what I just described here is what I would call a compositional solution to addition. It's a solution that relies on the fact that numbers are composed of digits. And leveraging this fact, we then came up with a finite set of recursive rules, which we're applying to a finite set of digits. And what's remarkable about this simple rule that you learn early in life is that it works no matter how long the numbers are. It can be 2-digit numbers, it can be 20-digit numbers, it works. It's absolutely remarkable. How do we model addition with neural networks? So my lab has worked on that for many years, so I'm one of the labs that have proposed models for this. And what I can tell you right away is that they do not treat numbers as compositional. Okay? Typical network for addition looks like this. And here numbers are represented as hill of activity across an array of neurons that are tuned to numerosity. Okay, so the neurons here, they care about when there are like 3 or 4 items on the screen. The one over there, they care when there are about 30 items on the screen. And number 13 here is encoded by virtue of triggering a hill at position 13. There are neurons in your brain and the brains of animals that are indeed tuned to numerosity. To be precise, typically you have Gaussian tuning curve to the log of numerosity. Been found in monkeys, and given what Lars said yesterday, I suspect it's true in bees. Okay, so here number 8 is encoded in the same way. And now this is how you're representing number. To add those two numbers, you need to create a hill at the top here that peaks at location corresponding to the sum of 13 plus 8, which is 21. And if you think about this problem, it's actually a nonlinear problem once you're working with those hills. And so you need an intermediate representation here, which you can wire by hand or train through backpropagation. Either way, you're going to end up with a smooth lookup table with nice interpolation properties and almost no extrapolation. So what I mean by that is that if you train this network with some pairs between 0 and plus 0 and 99 plus 99, you'll do fine with 1 and 2-digit number addition. But you would completely fail to generalize to 6, 7-digit numbers. Okay? And that's the problem with this kind of approach. We just have lookup tables. Now, this is just an example. There are many aspects of cognition that you can think of from that perspective of compositionality. And the prime example is, of course, language. As you know, language allows you to express an infinite number of sentences by combining a finite set of lexical items according to a finite set of grammatical rules. Right? And that's what gives its power to language. We can express any thoughts with a finite set of rules and words. But that's not just language. As I said, many aspects of cognitions can be thought from that perspective. So for instance, vision. You can think of visual scenes as being composed of objects along with their properties, in this case, their depth. In which case, scene formation is really a compositional process of combining those objects together according to the rules of image formation. And then vision should be thought as an inference problem of inferring which objects are in the visual scene along with their properties. And people like Anne Trisman talked about that like 30 years ago and talked about object files, if you look back at this literature. So it's evidence that we have such things in our mind. Okay, and it's a very active field of research still in vision today. It's not just deep learning. Some people are actually thinking about these issues of how do you represent visual scenes. So you can generalize the arguments to audition and touch and other aspects of cognition. And in fact, that's been known in cognitive science for years. 15 years ago, Josh and Bam came to this very conference and made this point to this community, trying to alert us that we need to start thinking that way. In fact, here's the last slide that he showed us at the time. He said, look guys, computational neuroscience is stuck in the 50s and the 60s. You guys are electrical engineers who work with concepts such as electrical circuits, optimal filter, control theory. Nothing to sneer at. This is the kind of science that got us to the moon. This is literally how we built rockets. But what he said is that if you want to model higher cognition, you need to move on. You need to borrow theories inspired by computer science from the 80s. Concepts such as compositionality, complex data structure, which I won't talk about, that's a big topic, pointers, simulators, and so on. So what Josh was saying is that, look, you guys look like this, engineers from the 50s, and what you have to start looking like is this, good computer science from the 80s. And Josh, being the leader that he is, he led by example. So he started his career looking like this, and he now looks like this. So that's what we have to do. All right, so Josh is not the first one to have made those points. In fact, those arguments have been around for 30 years. The very first talk I heard in my life as a scientist was from Jerry Fodor. And Jerry Fodor was destroying neural networks at the time. I will come back to this in a second. But he became particularly famous for this book, The Language of Thoughts, in which he pushed this argument all the way to the end. And he said, look, thinking is a fundamentally compositional process. It's about manipulating a finite set of concepts, or what he called mentalese, according to syntactic-like rules in your mind. That's how you derive new knowledge. And he pushed it all the way with symbolic systems, and I'm sure you all guys all disagree. But I think there was a fundamentally interesting point, nonetheless, in this book. So as I said, Jerry went a little further and said that, look, artificial neural network, at least what we call connectionist network at the time, which haven't changed that much since, are inherently enabled to be truly compositional. People like Dana Ballard made similar points, and were arguing that we need to go beyond this just simple architecture. And I'm not going to go into the arguments. If you read the Pilishin-Fodor paper, the original one, good luck. It's not an easy read, or if you read any of Jerry Fodor's book, it's pretty impenetrable. But the gist of the argument is the same as this one, is that in neural networks, we do not represent variables in a compositional way, in the same way that this does not represent number in a compositional way. Okay, so what was our response as a community to all those challenges for the last 30 years? How are we going to model compositionality? The bottom line is that we basically ignored the issue up until about 10 years ago, completely ignored the issue. If you review the literature on neural model of cognition, okay, 90% of them boils down to this: two neurons inhibiting each other's. Okay, I'm not making it up. Look at the literature, and I'm guilty as charged. I have published many models like this. Okay, in my lab we generalized to line attractor. Oh, big deal. It's exactly the same class of model. Okay, and if you don't believe me, just look at this week issue of Nature, and there is a model exactly like this in there. Okay, so it's beautiful work, but we have to wake up to the fact that this is what we do. We have reduced 70 billion neurons to 2, well, 3 if you count the inhibitor neurons. Okay, some people have tried to actually look at compositionality, such as Smolensky, Tony Plake, Chris Eliasmith, Alex Grave more recently. It's a bit strange because those models never really take off. They're published and then people forget. And nothing really happens because they never really fit what we see in the brain. That's been the main issue. And so I was personally losing hope. For 30 years, when I was in grad school, late 80s, I hear Jerry Fodor says, you guys are on the wrong track. And I've been thinking all my career, I want to work on this, but I don't know how. So I kept asking my colleagues, is there hope? What are we going to do? And the only argument I ever got from anybody, and many of you are in this room and you know who you are, we just need to scale up, Alex. It's just the architecture is just fine. As it gets bigger, it's going to work. I always thought that was the dumbest, laziest argument I've ever heard in my career, right? We were like faced with a really tough challenge, and all we could say was like, wait, and just scale up. Okay, well, a large language model came around, and I must say, I was like, what is this? And then GPT-1 appeared, predicting next word, 100 million parameters. You never heard of GPT-1. Why? Because it sucked. Okay, it wasn't very good at what it was doing. But then GPT-2 came along. Now we're talking a billion parameter, of the order of a billion. And I remember looking at it and thinking, whoa, that's a whole different ballgame. That thing is doing amazing stuff with language, even though it wasn't that great. But then of course, GPT-3 came along. Now we have GPT-4 and GPT-5 is on its way. And we have a whole zoo of those models now with incredible performance. So much so that now you can find papers like this one, an archive that many of you probably know by Sebastian Bubeck at Microsoft, where they talk about sparks of artificial general intelligence in GPT-4. So again, some of you probably have seen this prompt. If you haven't, it's really incredible. They ask GPT-4 the following thing. Here we have a book, 9 eggs, a laptop, a bottle, and a nail. Please tell me how to stack them onto each other in a stable manner. Okay? And here's what GPT-4 replied. Put the eggs on the book, arrange the eggs in a 3-row with space between them, make sure you don't crack them. Place a laptop on top of the eggs, and so on and so on. I'm going to stop there because what's amazing in this is the thought that GPT-4 thought of forming a 3-by-3 array of eggs. I have to admit, when I heard the talk, I didn't think about that solution when he showed it. So I was incredibly impressed that GPT-4 right away came up with this. I think it's stunning. Not everybody agrees, but I think it's really stunning that we are on this point in time. Here's another paper I really like by Andrew Lampinen at DeepMind. This is one in which he noticed that indeed GPT, in this case I think it was 3, maybe 4, fails on standard syntactic tests. It doesn't seem to be fully compositional. But what he pointed out is that if you test human in exactly the same condition as you test GPT, you get very similar errors in terms of compositionality, indicating that we seems to be in the right ballpark. It seems to be not far off from what humans are doing with grammar. Okay, so not everybody's happy. This is Noam Chomsky, the godfather of modern linguistics, who wrote an op-ed in the New York Times last year in which basically said, ChatGPT, fake news, nothing to do with language, this stuff is a fraud. I read it. I have immense respect for Chomsky, who I think have done absolutely amazing contributions to the field of cognitive science. But I can't help it and think that he's looking at this glass and he continues to argue it's 5% empty, when I think it's not unreasonable to argue that it is 95% full at this point. Oh, by the way, I know what you're thinking. Well, he looks suspiciously like an electrical engineer from the 50s. Okay, I want to reassure you, today he looks the part now. All right, so in the lab, we started thinking, okay, let's start looking into this. And along with Ryder Rivlin, a grad student in the lab, we started looking into how we could use those language models to understand how to use verbal instruction to instruct a neural network to perform a task. I should warn you, this work was started 4 or 5 years ago, and so at the time, that's all we had. BERT was kind of like state of the art, okay? So I let you imagine how much better we would do with the latest generation of large language model if only they could give us the weights, which is a bit of a problem for most of them. Okay, so our starting point was the observation that if you train an animal on the simplest discrimination task you can think of, it's going to take typically thousands of trials before the animal finally gets it through reinforcement learning, right? But in humans, I can take any human subject in a room, tell them, A, responds in direction of the stimulus presented with highest contrast, okay, and you show them this, and they know right away they have to respond in that direction, right? So that's called zero-shot learning. We take that so much for granted that we completely forget that by virtue of being in the instructions, we can completely bypass the learning process, right? We effectively rewire our circuit in our brain to do the appropriate sensory motor mapping without any training whatsoever. It's absolutely remarkable. And I always wanted to try to understand how we did this. So we started training. He started training an RNN on a variety of sensory motor mappings. In fact, we had 45 tasks. Okay, and on the side here, we plugged a large language model that could take the instructions corresponding to the task. So then when we train on the task in the context of having been told what to do. So we can do that. It works quite well. Okay? But here's a really interesting test. Now we take a brand new task that the network has never seen, and we tell the network what the task is about, and we ask can the network perform the task right away without extra training. All right? And the tasks we're using are fairly simple psychophysical tasks, like the one they're used in animals, but also in humans. Like an anti-go task here is response in direction opposite to the stimulus. So you see this, the stimulus appears, you respond in the opposite direction. For each of the tasks, we're going to have 15 different ways of describing the same task. Why? Because we want the network not to lock onto specific formulation or specific words in the instructions. Okay? Here's on the other extreme of difficulty another task called conditional decision making. Responds to the strongest stimulus only if you are confident, otherwise do not respond. So in this case, here's a display. There are two stimuli. Clearly, one is higher contrast than the other, so the network has to respond to the right in this case. Here's another display. Two stimulus appear. You cannot see them. They're about the same low contrast. And so here the right response to the networks is not to respond. Okay, so what's compositional about this particular task? Well, there are two aspects of our task here that's compositional. The first one is that the mapping from instruction to semantic has to rely on the compositionality of language. If you don't understand language is compositional, good luck extracting meaning from sentences. The second one is that our task set is an expanded version of one that was proposed a while back by Yang et al. In 2019, which we expanded, in which many of the tasks are actually composition of other tasks. So for instance, an anti-decision-making task can be thought as being the combination of decision-making plus anti-go. All right, so that's the problem that the system has to solve. Here's the gist of our results here. What you're seeing here is a learning curve in black here, which is the performance of the network on a brand new task as a function of number of exposure to that brand new task. Okay? Of particular interest is the first data point on this curve, which is a zero-shot learning performance. No training whatsoever. You're just being told what to do. And as you can tell, we're at 83% zero-shot performance. So on most of the tasks, actually, our network get them right away on the zero shot. Okay? And for some, a little bit of training, and as you can see, very quickly it saturates at 100% correct. I should mention that the large language model we're using here is SBERT, about 300 million parameters. If you use GPT-2, which was a billion parameter, 1.5 billion for the largest version, it doesn't work well at all. So it turned out that it really matters which language model you use and how you train the language model. That's really what matters. So all those details are in the paper that's coming out soon, but you can see the bioRxiv version. Just so that you know, the blue curve here, that's how well you would do if you had no language whatsoever in your network. That's kind of like lower bound on performance for a non-linguistic network. So we're doing way, way better. So what you just heard here is, I think to my knowledge, the first model of how Wernicke's area that's responsible for language comprehension can reshape circuits, sensory motor circuits, on the fly according to instructions. Okay? And if you want to understand a bit better the compositional aspect of this RNN, you should really go see the paper by Driscoll and Zhixiao, and David Zhixiao will give a talk about this, I think, later in the conference, where those guys have started to unearth how this network here is indeed compositional and how it encodes the different elements of those tasks. So we've done Wernicke's area, but how about the reverse? Can we also model Broca's area? Broca's area does something that's completely amazing. It gets spikes from neurons that are involved in sensory motor transformation performing tasks, and it turns these spikes into verbal description of how the system is solving a problem, which I found just as equally fascinating as the other direction. So we worked on that. It's very preliminary stuff, but what we did is we had network perform a task. We could train it with whatever, reinforcement learning, no language. It figures out how to solve a task. And here we have a decoding network that's trained to describe with words which task it is that this network is performing. So the network might say response with the identical orientation when prompted. So we tried that, and we can get the network to generate instructions. But how good are those instructions? So you could just like rate them by hand. But we thought there is a cooler way to really test this, which is that you can feed those instructions in another network of the same type and see whether it performed the task, right? And so we did that, and long story short, you can go see the results over there. Basically, the second network here performed the task nearly as well, whether the instruction comes from us, and they're valid instructions, or whether they're generated by another network that's performing this task. So in other words, we got networks, two networks to communicate with natural language here. So the beauty, of course, of having a neural model is now we can look inside and try to understand how it solved the task. So here, we're now collaborating with two groups, one at the University of Geneva and one at Columbia University, where we're doing fMRI and intracranial recordings to start looking into the neural basis of compositionality. And I don't have any results to show you yet, because this is really ongoing work, so hopefully next year we'll present this at Cosyne. So that's the first project I want to tell you about. I'm going to tell you about the second one very briefly. This is one with Pablo Tano, another grad student in the lab. Pablo will actually give a talk this afternoon on this project, so that's why I'm going to be brief. This is one in which we were interested in learning compositional representations. So Pablo did that in the context of navigation. So imagine this animal is trying to go to this reward here and has to go around the wall here. So how do we solve that with reinforcement learning? Well, basically, the animal will have to take a step, consider its option, values of different options, take another step, and repeat and repeat and repeat, and eventually get to the rewards. Then use TD learning to optimize the trajectory. Very long, takes forever. So what Pablo was wondering is there a way to decompose complex behavior like this as being the composition of several smaller, simpler behavior? So for instance, you can imagine that a behavior like this is really two trajectories, one primitive, a behavioral primitive like this, and a second one like this that gets you to the reward. If you could learn that way, perhaps you could go a lot faster. And indeed, this is what Pablo is going to show you, that if you have a combination of predictive representations with an adaptation system, or what people in control theory would call a forward model with an inverse model, and you concatenate those things together, you can, through gradient descent, very quickly to learn to decompose a behavior as the sequence of behavioral primitives. So my argument here is that, yes, we can start studying the neural basis, and in particular in humans, because that's, of course, the easiest target for this kind of functions. But the dream will be to do it in rodents, of course, because there we can perform such incredible recordings. So Tim Behrens and his lab has started to look into this. I'm sure he's not the only one in the community. It just happened to be quite familiar with this particular work. So I want to highlight how those guys are starting to tackle this in rodents. So what they did is they had rodents navigate a maze that's composed of 9 platforms that are linked to one another with little bridges here. And they trained the animal an abstract task, which is go to location A, then B, then C, then D, and then go and then repeat. Okay? And every time they get to one of those locations, they get rewarded. But the mapping of A, B, C, D onto the physical location here changes from block to block. So at the beginning of the experiment, for instance, here, the animal learns through rewards that A is here, B is here, C is here, D is here, and then they repeat that for a while, and then they change that on the animal, and suddenly the animal has to figure out that A has moved here, B here, C here, D here, and so on and so on, right? So you can see kind of like the computational nature of this work here. We have an abstract rule, like a syntactic rule. It's quite simple, applied to a finite set of position in space. And you just have to learn that mapping in different blocks. So it has exactly the kind of right flavor. So how would you solve such a problem? There are many ways you can solve this problem, but here's one way. What you could do is you could have in your mind a counter at every location. So the 0 here is meant to be a counter, okay? And then the animal is thrown in there and moves around, and as long as it doesn't get rewarded, nothing happens. And then, you have to imagine, the animal doesn't know where A, B, C, D is yet, right? It's trying to figure out where it is. And then just by luck, it gets to A, it gets rewarded. So at that point, we're going to activate the counter here. Just activate it. We're not doing anything else. Then moves around, and it gets lucky, it gets to location B, gets rewarded, activates a counter at location B, and then it increments the one at location A. Okay? Moves around, nothing happened, no reward. Oh, it got to C, it's rewarded, activate the counter there, increment the other two. Moves around, got lucky, got D right away, increment, activate the counter over there, and increment the other ones. At that point, the animal is done. From there on, all it has to do is figure out where does the counter say 3? And that's where it should go. Oh, it says 3 over there. OK, let's go over there. And that is the right answer. That's A. When it gets to A, it resets the counter to 0, increments the other one, and just looks for wherever it says 3 again. And that just happens to be location B. So the animal is done if it has that. And if you go read this paper, you'll find that more or less, I'm looking at Tim here, and I'm very nervous. That's more or less what those guys have found. In fact, it's more complicated. I simplified it here. There's more to it. But that's a gist of what those guys have found. If you look in the MPFC, medial prefrontal cortex, there are neurons that seems to be incrementing those counter with line attractors. Beautiful stuff. To me, I have to say I love it, but it's a bit brute force. It's basically a gigantic lookup table that scales with the number of states that you can visit. There are only 4 rewarded locations. Why is the animal bothering encoding all the location space? Imagine this is now a space of like 1 million possible states, and only 3 are rewarded. That seems to be a bit brute force. But you often find that when you train animals extensively on a task. They start covering all their options. So I'm not so surprised. I also would like to give you a pointer to this beautiful paper by James Whittington, who works with Tim, that's on bioRxiv. And when James showed that this kind of approach, those counter, what he called memory slots, actually allows you to explain a wide variety of tasks. It's absolutely gorgeous theoretical work, if you ask me. And also, you should go see the poster of Jacob Beckermans and Behrens that's at this conference, where those guys are also exploring all the solutions involving the hippocampus and grid cells, because that's Tim, and trying to argue that this is how you really you can do something that's more compositional in a sense. But absolutely gorgeous work. I think those guys are leading the way, and you guys should really check this, because this is, I think, the way the field is going to go in the next decades. More generally, I think we have to start thinking about more complex tasks of this sort. We have to start creating an environment in which a task is composed of other tasks. Some of you may be familiar with the work of Bob Dada, where he's actually thinking of natural behavior as being a sequence of smaller syllables. He talked about this, I think, last year or a couple of years ago. Beautiful work, and I think that's exactly the direction in which we should go. And then the goal will be to really understand when an animal sees a task, how does the animal infer some compositional semantic representation of those tasks? How does it decompose it as being other subtasks that are combined according to some grammar? And in a way, that's very much what Fodor was talking about with the language of thought. Of course, he was thinking symbols, regular computers, that's not how the brain works, but I think the spirit is the right one. And I think that once we do that, we'll be a step closer to understanding how language interacts with cognition, because for me, language just plugs into this pre-existing compositional representation that you already have in animals, possibly in bee, I don't know, but certainly in rodents, and that after that, maybe it's just a question of scaling up. Because that's really the key, is understanding that thing here. All right, so 2008, Josh was on this podium saying, you guys have to work on that. And for 15 years, we did nothing. But I think that we are there. We can finally do it. And I just give you some examples. There are more examples of using this architecture to understand, of course, for instance, the neural basis of language. Beautiful paper by Martin Schrimpf on the topic. And guess what? Look at who is a co-author. So, Josh didn't wait for me to tell him that something is happening. He's himself actively engaged in this kind of research. OK, I want to finish on a note of caution. If Randy Gallistel was sitting in this room, I can tell you he'd be already screaming at me in a very nice way, because Randy's a very nice person, but very passionate. And he'd be like, Alex, you're completely wrong. There's nothing compositional about those systems. And I have to share with you a little dirty secret about ChatGPT. I was playing around the other day trying to get it to add numbers. Somebody asked a question about that yesterday, that ChatGPT doesn't add number well. Well, that's no longer true, people. You can try to ask ChatGPT to add any pairs of numbers. It would do it perfectly well. That wasn't true for GPT-3. But GPT-4 is perfectly fine. You guys are probably familiar with this thing here. If you click on this, it will give you an idea as to how ChatGPT came up with this particular response. If you do it with adding numbers, you get this window, and this window is Python code for adding number. It's incredible. The way it adds numbers is by writing code that it runs on a conventional computer, not by running it through the transformer. Okay? So I don't know what bees do, but I would guess they do not write Python code. But they're probably using one of those neural networks that I showed you before that are not compositional. Okay? But ChatGPT just cheats. It's a clever solution. I'm sure like Randy would get a good chuckle out of that one. And it's still going on. There is a paper that just came out last month from DeepMind on solving geometry problems with AI. It's hybrid systems. It's a neural network paired with a symbolic system, just like for AlphaGo, by the way, and all those programs. And many people are arguing, of course, in the field that, LLMs is not the solution to compositionality. We'll never get to general intelligence with this. This is a recent paper. If you follow Yann LeCun every single day, you're bombarded with 3 tweets saying that it's not going to work with LLMs. Okay, we get it, but I do think it's a good starting point, okay? And to quote Churchill, I'd say, okay, this is not the end, it's not even the beginning of the end, but it is perhaps the end of the beginning. So on that note, thank you to, first of all, the members of my lab who have been involved in the work that I showed you, Pablo and Ryder, and all the people who have really inspired me over the last 30 years to think really hard about this problem. I think it's really amazing that we're finally there. Also, big thanks to University of Geneva, which is crazy enough to actually give me the funding to do this, because I can't find funding so far for this. And if you like this kind of topic, you want to work on it, good news, I'm hiring. And even better news, my lab is an hour away from this. So please apply. Thank you very much.
So if you have any question, please come to the microphone.
Alex.
Yes.
So it's interesting to me. I'm going to try and be as quick as possible. I'm not going to try and stand in for Randy Gallistel, but I don't buy the story at all. Okay.
Randy's story or my version?
Yours.
Oh,
yours.
Mine. Ouch.
Yeah. Okay. So just a couple of things. It's interesting to me that you mentioned Josh, and then you showed sort of, we have Wernicke's area. You know, Josh and Ev Fedorenko have a paper where they make a clear distinction between cognition and language, or what they call linguistic competence and functional competence. And one of the main points that Ev makes is that Wernicke's area is not cognition. Okay? So, but you can understand syntax and you can arrange words properly. So I think it's kind of ironic that Josh and Ev and others have a paper where they make it very clear that language areas are not actually thought areas. That's one.
Okay. Can I reply to this? Because my memory is too old to remember 2 points. I think you're making a mistake. What they're arguing, indeed, is that language is not thinking. But the argument I'm making is that thinking is compositional just like language. And I think Josh would agree with me. That's a different point. Both are compositional, but they're not the same thing.
Yes, but in your experiment where you have the one-shot learning, I would argue that a human does one-shot learning because they get and understand what's going on. And I'm saying that you can fake that in simple tasks just with linguistic competence. They're not understanding. And then the second point I like to make is with respect to Tim's paper, right? So Tim and I had a back and forth about that paper, which I do think is fantastic, obviously. But he admitted to me that the actual representation of the ABCD, the actual abstraction, could not be found, right? In other words, there was a... well...
For those of you who haven't heard, Tim is saying this is totally untrue.
I'd like to see, we can record it back. Is it actually true that rather than the brute force approach, there's an actual representation of the higher level abstraction. Oh, it's not there in the mouse. It's not there in the mouse.
Entorhinal, that's what he's saying. It's in the entorhinal.
Just to clear what
Tim has a bit of an obsession
just
with
to clear
grid
what
cells,
Tim
so
just
this is where
said,
he wants
the
to find
experiment, the paper, just to be clear, the paper was in the mouse, and they did not find it in the mouse. They found it in the primate, and now they're looking for it in the mouse.
Okay.
Let's see if they find it.
You've had the microphone long enough anyway.
I actually enjoyed the larger point, so thank you for the talk. So I'm afraid I'm going to come across as a little bit of a sort of glass 5% empty person with this question, but it's really meant as a genuine question. So I noticed when you were describing the instructions you fed to your network, you didn't just feed one linguistic description of the task. You said you fed 15 or something like that, right? And this is a general property of transformers where you can get pretty dramatically different results with very small changes in the prompt. On the flip side, there's actually techniques for essentially extending the token length of a transformer by compressing prompts into what to a human looks like complete gibberish, but the network understands perfectly. So my question is this, right? I mean, to what extent is that basically signaling that these networks are again, and this is actually related to the previous question, they don't have a clear understanding. In other words, they're very brittle in their understanding of language, and to a certain degree, are more brittle, let's say, than a human because it's not fueled by other or constrained by other cognitive models? Or is this, again, just a matter of let's just wait for the bigger network and it'll solve this problem because we'll regularize better or something like that? So I'm curious what your thoughts are on the fact that you have to do this.
I mean, yeah, to some extent, it's beyond what we're doing because this is really a question about GPT. But I've heard this argument for 2.5 years now, and people saying, well, it's brittle. So I don't know, GPT-4 for me, it's good enough. I mean, like, you really have to go out of your way to get it to do something silly and stupid. I mean, it's the linguistic ability of the system is really remarkable. I'm sure you've tried it. So I don't know, this is not me, I don't develop GPT, but I feel like it's my message to you guys is that it's more than good enough to look into the null basis of compositionality in animals. I don't know about language.
Thank you.
I have a question. You said that there's the remarkable ability of humans to use verbal instructions instead of operant learning, to sort of make a shortcut. Do you know if there are any studies that compared whether the learned behavior is indeed identical? Let's say if you do learn it with one verbal instruction versus trial and error of 1,000 trials, will you make the same mistakes? Are there complex enough tasks that can expose this?
I don't. But I think that we should start thinking that way. And I also think that, look, those tasks are very simple, the one we're using here. We're starting to think about this, Ryder in the lab is starting to think about this, is how do we actually also discover structure in a task? And what's very likely that we also learn through hypothesis testing, which is not what RL does currently. RL is just like small gradient descent, right, in the parameter space. But I think as you start thinking compositionally, you're going to have to rethink learning as being more about testing different hypotheses that correspond to the task that you're currently looking at. And we're just starting to look into this, but I don't know of any good studies on that yet. It's hard to study that, learning that way. Randy Gallistel talks about that at length in his book, by the way, how we learn through hypothesis testing.
Thanks.
Hi there. I'd like to engage a bit more critically with the example that was provided yesterday. So it seems like much that compositionality is going to afford us is to achieve things that, for example, a cross-mode compositionality. Yesterday, they talked about the ability to render an image with 4 individuals, but then being returned one with 5 or 6. So adding 2 things can be solved with Python code. Counting individuals in an image is a bit more involved, and it doesn't seem like we're there yet. So can we really say that we're 95% of the way? And to what's that benchmark that you believe we've hit that encourages you to offer an estimate that's so high?
Yeah, no, I agree. There's a bit of a cheap shot at Chomsky, the 95%. That's why I want to end with the note of caution. I hope Blaise will talk about this. Blaise Aguirre is talking this afternoon or later this morning, and I know he's been one of the people arguing that general intelligence is just years away, months away, that would be 95%. I'm less optimistic. I do think that we're missing some key ingredients here. So I will gladly acknowledge that, especially for counting. One last, or we're done?
We are going to have one last.
Okay.
One last.
Alex, fantastic set of ideas and very challenging.
I cannot hear you very well.
Fantastic set of ideas and very challenging to think about them, but aren't you cheating a little bit by moving the compositionality problem straight into the ChatGTP? And a lot of the interesting things, a lot of the interesting computations are actually going to be outside of your networks. Should we try to open up ChatGTP, say, hey, is there any type of representation similar to that, what we see in different brain areas? Or should we try to think of a more neurorealistic ChatGTP?
Come and hear David Susio's talk, right? That's why I was trying to point out the RNN itself is compositional. And if you ask me, the microcircuit in cortex, whether it's in V1 or in Broca's area, has to be compositional as well. So I think we have to think just along the lines of what you're suggesting. We have to really start going beyond this architecture and even try to understand what is it that makes microcircuits in cortex so good at compositional computation.
All right, thank you, Alex. All right, our next speaker is Margarida Sousa.
Good morning, Cosyne. So today I'm going to tell you about our discovery that dopamine neurons encode a multidimensional probabilistic map of future rewards. Can you hear me? Sorry. Yeah. Okay. So interacting with the environment through the interactions with the environment is at the core of reward processing, and it's thought to guide adaptive behavior. So, for example, a mouse foraging in a forest for food is faced with a decision between two potential food patches. The mouse might store a single quantity that reflects previous experience, for example, the average collected rewards in each patch to guide future decisions. This is a very simple strategy, but however, it discards perhaps useful information about reward variability in the environment. So recent work on reinforcement learning that learn about the average expected rewards have been generalized to learn about the full distribution of rewards in the environment. This innovation is termed distributional reinforcement learning. So instead of learning one value, a set of values are learned through biased updates that weight asymmetrically positive and negative reward prediction errors. So for example, an estimate that is more sensitive to positive errors will converge to a positively biased estimate, and vice versa for one that is negatively biased. For the case of our foraging mouse, through learning, these values converge to statistics that provide information about the full distribution of rewards in the environment. Distributional RL has been shown to improve representation of the environment in deep RL tasks. In the brain, recent studies have shown that midbrain dopamine neurons long thought to encode these scalar reward prediction errors are implementing a similar algorithm. But Let's work has focused on A, understanding how this information is used to guide behavior for action selection and decision making, and B, to understand how these value learning approaches can be generalized to other dimensions over which rewards can vary. For example, rewards can also be distributed in time. And standard reinforcement learning approaches are estimating a value that compresses information about magnitudes in time and therefore discards information about reward times. So the work I'll present to you today is generalizing the core idea of distribution RL to the time domain by considering a diverse set of temporal discounts, like represented here, that discount future rewards heterogeneously and allow for extracting information about the distribution of rewards in the environment. And by combining the tuning to reward times and magnitudes, we have access to this joint distribution over these two dimensions, a kind of non-spatial probabilistic map of rewards in the environment. So if animals or artificial agents were given access to this map, this might afford significant improvements in face of dynamic environments, as I'll come back in the end of the talk. This is a short talk, so please read our bioarchive if you're interested, and also the bioarchive from Paul Massett and Pawel Bujalski, showing complementary work on the topic. Okay, so to test for signatures of distributional learning in time and magnitude, we performed a series of electrophysiological recording of optogenetically identified dopamine neurons in the midbrain. Our task is a simple classical conditioning task where other cues predict rewards. I'm going to start by describing our attempts to reproduce previous results that show that dopamine neurons encode the distribution of reward magnitudes. So we're going to use the trials in which the cue that predicts a variable reward magnitude was given, sampled from a slightly bimodal distribution. So here's a single dopamine raster aligned to the reward delivery. Trials were sorted by reward magnitude. The bigger the reward magnitude, the more this neuron fires. It has a zero net firing rate at about 5.4 microliters. But as has been shown before, if we look at these reversal points for the entire population of dopamine neurons we recorded from, we see that there's diversity. And this is a proxy for the diversity in the sensitivity to reward magnitudes that is a core feature of distribution RL in magnitudes. So then by taking into account these reversal points and these symmetries for rewards on the left and on the right, we can decode, as had been shown before, the distribution over reward magnitudes. Okay, so this is a nice sanity check, but what about time? Are distributions of reward time encoded in these neurons? So we added 4 extra cues that predict a certain reward amount after variable delays. And we looked at single neuron responses to understand if they indeed discount future rewards compared to immediate one in a diverse way. First, we just checked that mice learned to use orders to predict rewards at different times, as can be shown here by the time course of the anticipatory licking. So here's an example raster of a dopamine neuron aligned to the cue delivery. Trials were sorted by rewards delay. We can compute the mean for each condition and estimate the temporal discount, here represented as the parameter gamma. We can do this for all neurons that we recorded from. So for example, this neuron here on the right discounts the delay to reward in a weaker manner. So essentially, it cares about rewards in a longer time scale. And now if we look at all neurons, we can test our first prediction. That was that there's variability in these temporal discount factors. So here, each curve is a fitted temporal discount function. We can see that it seems that there's variability in the temporal discounts, and it doesn't reflect noise, because if we divide trials in two, the estimated temporal discounts are significantly correlated. But we wanted to understand if this diversity contained information about the distribution of reward times. So I'm just going to briefly explain by giving a simple example why this would be the case. So here's a schematic. So each curve here is a temporally discounted value at a cue that predicts rewards at different times, as represented here on the x-axis. And the idea is that if, for example, we look at a population vector activity for a queue that predicts imminent rewards, then we see that all channels here reflect that this queue happened with more or less in a greater or less degree. But now if you look at the population activity for a queue predicting later rewards, we see that only the channels that weakly discount reward delay reflect the occurrence of the queue. And this difference in the shape of the population activity is what allows to decode distribution over reward times. So what about real data? So here I'm showing you the decoded densities using the 43 dopamine neurons we recorded from for the queue that predicts immediate rewards after 1.5 seconds, 3 seconds, and 6 seconds. So indeed, the system contains information about the timing of future rewards, but how does this information relate to behavior? So we leverage on the fact that animals show variability in the anticipatory licking rate to compare the decoded time from this population of dopamine neurons with the time course of anticipatory licking. So basically, we divided trials in half, where animals starting licking early or late. And we see that for the queue predicting rewards after 1.5 seconds, when we again decode the distribution over reward times, there seems to be a qualitative agreement where decoded reward times from early and later licks are shifted from earlier to later times. And this qualitative agreement seems consistent across the other cues. So this represents evidence that the reward time we're decoding from the population of dopamine neurons is similar to the reward time animals are using to guide when to start licking. And importantly, this information was decoded at the cue, at the onset of the behavior episode, where it might be useful to drive behavior during that episode. However, previous accounts of distribution RL in magnitudes have shown that information of the distribution over reward magnitudes was only shown at rewards, at the end of the behavior episode. But in principle, this information should be backpropagated from the ends to the beginning of the trial. Well, if we look at this variability in the sensitivity to reward magnitudes, as measured by reversal points, as a function of the firing rate at the cue, we don't see a clear relationship. But interestingly, if we correct for this diversity in the temporally discounting of delays to rewards, then we can see this hidden relationship between the variability in the coding of reward magnitude from the responses at the reward to the responses at the cue. And this is really cool because it allows us to decode the distribution over reward magnitudes not only at the end of the episode, at the reward, but also at the cue, which would be useful to potentially to drive behavior during the behavior episode. So I showed you independently that you can extract information about reward times and magnitudes from the responses of this population of midbrain dopamine neurons. In our experiments, reward times and magnitudes are independent, so if we factorize the joint distribution, we obtain these two-dimensional probabilistic maps over reward times and magnitudes. This is for the queue predicting a certain reward amount. Now we compare it with the queue that predicts a variable reward amount, and we see that these maps can indeed reflect the difference in the variability in reward magnitudes. And this seems true for the cues predicting also for the time, when you look at the maps for the cues predicting rewards at different times. This map of future rewards was decoded using the activity of only 43 dopamine neurons over an interval that is shorter than half a second. We think these maps were learned using a simple modification of a local in time temporal difference learning algorithm. But surprisingly, they allow for this very rich and complex representation of rewards in the environment. So now I'll finish by giving you a sense how these maps can be used to drive animal behavior. So now, going back to our foraging example, we ask how can having access to this map improve behavior? And the idea is that having access to this map, if the animal only had access to a scalar value, then he could end up going only to the later larger reward here on the right, to the right patch. But if he has access to this map, then he can make a better use of his time and go first to the early reward that is smaller on the left, and then to the later, bigger reward on the right. And in the end, of course, collect more rewards. So here I'm showing you simulated behavior from two agents, one that has access to this scalar value to guide behavior in pink, and in blue, one that has access to this time magnitude map. And we see that indeed, the agent that has access to this map can collect more rewards. So I would like to thank everyone involved in this project, so Pawel Bujalski and Bruno Cruz, who collected the data and were very important in the design of the task. Our collaborators, Daniel McNamee and Kenway Louie, my PhD supervisor, Joe Paton, my lab, especially Teresa Duarte and Simon Zamora, that helped me a lot during the presentation, and the funding.
Okay, we will have one or two questions while the next speaker is setting up the computer. All right, I have a question actually. So when you were showing that, the plots of the probability versus the time until the reward, was that so? That's not really from the neural data, right? That's like from the experimental setup.
Which plots?
Yeah, that, like the reward probability versus magnitude. So that is actually from the experimental setup, or is that from the neural?
No, this is from the population activity of neurons, dopamine neurons. Yeah.
Hello? Hi, sorry, I don't actually have a question. I realized that it's always disappointing when the audience don't give questions, and this was a really beautiful talk, and I just wanted to say it was stunning. Thanks.
Thank you.
Hello? Hello? Can I had just a quick question. Any idea where those dopamine neurons are getting the timing information? I mean, do you think that's an intrinsic biophysical mechanism that has them counting, 3 seconds, 6 seconds, that kind of thing?
Yeah, so we think based on Ganglia as a reinforcement learning system that receives information about the state of the world, maybe time, and then is estimating the value given these reward prediction errors signaled by dopamine neurons. So we think that one of the hypotheses is that maybe it's not the temporal discounts of the tuning of the functions that change, but it's the inputs or the time that they get as inputs that is different. So yes, that's a possible hypothesis.
I was just wondering where the time information in the brain is being calculated, like the clocks that are doing 1 second and 2 seconds and 3 seconds and 6 seconds, and how far that goes.
Yes, we think that maybe, yeah, that representation can influence, and maybe different neurons have access to different time windows. Yeah.
Thank you. Before we get started, the last speaker of the session, if you are here in this room, could you come to the front? Because, yeah, all right. Yeah, yeah, cool, awesome. So our next speaker is Zhixiao Su.
Thank you, everyone. I'd like to apologize for my voice first. It took me too long to recontrol it after being COVID. So today I'm going to share with you how locus cerebrus norepinephrine neurons contributes to learning from errors. We animals live in a dynamic world. We take actions, learn from feedbacks, hoping to do better next time. This can be realized by the brain through reinforcement learning. It makes decisions of actions according to decision variables and update these variables according to the discrepancy between reward and prediction of reward, which is called reward prediction error. This type of error signal can be broadcasted across the whole brain through dopamine neurons in midbrain. However, although dopamine neurons innervates the basal ganglia densely, it does not go to cortex much. So how does cortex learn from errors? Here, the catecholamine sister of dopamine, norepinephrine, came to our attention. In contrast to dopamine, norepinephrine axons innervate the cortex a lot, but leaves basal ganglia mostly alone. And interestingly, these two systems share very similar postsynaptic mechanisms for neuroplasticity control. So is it possible that cortex learn from errors through norepinephrine neurons? We want to test this idea whether it receives error signals from locus cerebrus norepinephrine neurons. These neurons are located in the pons and project broadly across the whole brain. To answer this question, we first need to develop the behavior task where a mouse learns from errors. This is what we did. Here, a thirsty mouse is presented with two lick tubes. On each trial, it can decide to lick left or lick right and get rewarded probabilistically. The probability of getting rewarded on the two sides changes across time independently. So here, the size of the blue dots represent the reward probability changes across time, and the bottom I'm plotting a running average of relative reward ratio of right versus left. You can see the reward contingency changes multiple times across the whole session. So in this dynamic environment, what the mouse needs to do is to integrate our past information to make better future choices. So this is what the mouse actually did. The black ticks represent the animal's choices. The ones on the top meaning the animal chose right. The ones on the bottom meaning the animal chose left. The longer ticks meaning the mouse got rewarded, while the shorter ticks mean the mouse didn't get rewarded. You can see what the mouse usually does. It will mostly stay on one side when get well rewarded, while when the reward probability drops, it will not get well rewarded and switch to the alternative choice, and back and forth. You can see the trend of the choice in black follows while the reward trends in blue. To test if and how the mouse utilizes past reward information, I fitted the logistic regression model to predict the animal's future choices using choice history and reward history. So here, the regression coefficient is showing that past outcome information contributes to future choices, and the influence gets smaller as the history gets longer. This is consistent with the reinforcement learning agent who continuously updates his prediction according to new outcome information. So we fitted a reinforcement learning model to the animal's behavior. On the right, we can see the animal's choices in black can be well predicted by the model in green. So till now, we have found a behavior task where the mouse learns from errors and a reinforcement learning model that helps us to infer the errors the mouse is learning from. Next question is what norepinephrine neurons are doing in this task. To do that, I'm trying to recording from action potentials of locus cerebrus norepinephrine neurons. To do that, I implanted the electrode bundle into the pons. And then to identify if the neurons I recorded from are norepinephrine neuron or not, I specifically expressed the light-sensitive opsin into norepinephrine neurons. In this way, I will be able to tell whether they are norepinephrine neuron by whether they respond to light stimulus. On the right, I'm showing you one example electric trace where a norepinephrine neuron is consistently responding to the light stimulus on the top indicated by the blue bars. On the bottom, I'm showing you another two example norepinephrine neurons with their rasters. You can see from their raster that all of them are responding consistently to the light stimulus marked in blue. You probably have noticed that these two neurons have different action potential shapes, with the left one being more wide, but the right one being more narrow. This is actually the case we see across all the neurons. So in all the neurons we recorded from, they fall into generally two clusters, with the first type 1 cluster featuring wider action potential shapes, but the type 2 cluster features a narrower action potential shape. If we project them onto their first principal component, we can see they fall into two discrete clusters. The existence of these two types of waveforms is also confirmed using high-density electrode neuropixel probe recordings. Interestingly, type 1 neuron also tends to be more dorsally distributed compared to type 2 neurons. This phenomenon has been previously seen by our colleagues in rat. So what do they do as the mouse learn from errors? Let me show you two examples. Here are the raster plots of two example neurons, each from type 1 and type 2. The raster plots are organized trial by trial, aligned to the time of the choice, followed by a reward delivery indicated by the dashed line here. The raster plots are sorted according to reward prediction error on that current trial, with the darkest red represent the most negative reward prediction error and darkest blue represent the most positive reward prediction error. You can see if we focus on the outcome window, type 1 neurons activity gradually increases with reward prediction error increase, as is reflected by the average firing rate here. While on the other hand, type 2 neuron example has a stronger activity when there's negative reward prediction error, which corresponds to the absence of reward in this case. We can also see the trend more clearly in the average firing rate. So these two neurons have distinct responses to reward. So it is happening to all the neurons in these two different types. To test this, I fitted a linear regression model onto the spike counts in the reward delivery window with two major regressors, reward and reward prediction. Here I'm plotting the t-statistics of these two regressors for all the neurons. For one specific neuron, if its t-statistics of these two regressors falls into the first quadrant, it means it is positively correlates with reward, but negatively modulated by reward prediction, which is consistent with the definition of reward prediction error response. While on the other hand, if a neuron is located around the negative part of x-axis, it means it's negatively correlated with reward, with no significant modulation by reward prediction, meaning it responds to absence of reward. When we plot the actual neurons' t-statistics into this space, this is what we see. We see type 1 neurons clustered around the fourth quadrant, and type 2 neurons clustered around the negative part of x-axis, meaning type 1 neurons are towards the encoding of reward prediction error, while type 2 neurons as a population responds to the absence of reward. This trend is again captured by a polar histogram derived from the regression coefficients from the linear models. Here we can see type 1 neurons clustered towards the direction of reward prediction error encoding, while type 2 neurons are clustered towards the direction of absence of reward encoding. If we look at the populational average activity, we can see type 1 neurons activity gradually increases with reward prediction error increase, as reflected by a linear tuning curve with respect to reward prediction error. However, type 2 neurons activity is purely higher when there's absence of reward, as reflected by a step function shape here. To see if they actually are contributing to the animal's future choices, I again plotted the behavior policy update against the spike counts of these two neurons in the reward delivery window. We can see type 1 neuron activity positively correlates with behavior policy update, while type 2 neuron has a negative correlation. It means the more the type 1 neuron fares, the more the animal is going to repeat the current choice in the future. Till now, I have showed you we have found two types of norepinephrine neurons in locus coeruleus, with type 1 neuron clustered more dorsally in locus coeruleus and encoding reward prediction error, while type 2 neurons are located in more ventral locus coeruleus, responding to absence of reward. Because there has been previous anatomy work showing norepinephrine neurons projecting to cortex are more located dorsally in locus coeruleus, it makes me want to answer our original question: does cortex receive error signals from norepinephrine neurons? Here, from type 1 neurons maybe. To test this directly, I recorded from the calcium dynamics in the norepinephrine axons from locus coeruleus located in prelimbic cortex, where we have previously seen neural representations of decision variables in this dynamic foraging task. Here we can see the average activity of axonal dynamics gradually increases with reward prediction error, as is reflected in the tuning curve. If we again repeat the previous analysis we did to single neural activity to this calcium dynamics session by session, we can see the majority of the sessions fall into the fourth quadrant of the t-statistics space. And in the polar histogram, they point to the direction of reward prediction error encoding. This trend of distribution is very similar to what we saw previously with type 1 neural activity. So potentially, indeed, this reward prediction error signal is provided by the type 1 norepinephrine neurons. This is again tested directly when we record from norepinephrine dynamics directly using Grabenine sensor. Here, the norepinephrine dynamics also gradually increases with reward prediction error. If we repeat our previous analysis again, we can see the norepinephrine dynamics are clustered towards the first quadrant and point to the direction of reward prediction error encoding, similar to type 1 activity and also calcium dynamics of the norepinephrine axons. So now I have showed you two types of norepinephrine neurons in locus coeruleus, with type 1 neuron featuring a wider waveform located in dorsal locus coeruleus and activated by reward prediction error. It potentially provides this information to the cortical area for learning from errors. However, type 2 neurons features a narrow waveform located more ventrally in locus coeruleus and activated by absence of reward. We still don't know where type 2 neurons project to, but here let me show you the first few fully reconstructed norepinephrine neurons. If we color code these neurons by their projection specificity, it shows that the system is probably modular, with potentially subtypes of neurons exclusively projecting to brain areas like cortex, cerebellum, and medulla. Interestingly, the neurons projecting to cortical area are more dorsally located, so potentially they correspond to type 1. It is possible that type 2 neurons correspond to the other types of neurons with different projection specificity. Then how about dopamine neurons? We have previously seen these two systems have different projection specificity. It makes it very tempting to say that maybe there are two different systems learning from errors in parallel, potentially for different modalities and different scales. So with this, I would like to thank my mentor, Dr. Jeremiah Cohen, for his guidance and support, my collaborators, IFES team, and our anatomy team for their help, and our founder of the Institute, Paul Allen, for his vision, encouragement, and support. Thank you.
All right, we want to make sure you get your coffee reward at the predicted time. So let's just have one question while the next speaker is setting up the computer.
I guess. Sorry. Hello? I guess I'm the question. So thank you for your beautiful work and presentation. I actually have two sub-questions. So I wonder, how do we interpret that your recordings has a strong, like a pretty high magnitude signal to the goal cue?
Yeah, you're right. This is very consistent with what we previously have seen with norepinephrine neurons when they correspond to salient stimulus in the environment and also action, like engagement of certain actions. We haven't focused on this analysis yet because because this dynamic filtering task is more related to learning from errors. But some preliminary result shows that the first peak have something correlated with how fast the animal responses to the cues, to the go cue. Yeah.
Thank you. I have a second sub-question. I wonder, because I know very little about like NE as a learning signal, so I wonder, is there similar things like in the dopamine system, like if you keep stimulating like the NE neurons, the animal will be addicted, or?
We have tested the hypothesis by inhibiting these neurons as the animal learns from errors. We saw that when we inhibit the reward prediction errors, it treats the animal by a more negative reward prediction error. So they hate the current choice and prefer the alternative choice more. So that is consistent with the reward prediction error hypothesis.
Thank you.
Thank you.
Thank you. All right, our last speaker of the session is Martha Garcia Garcia.
Thank you. Can everyone hear me? Okay, so today I'm going to talk to you about cerebellar granule cell climbing fiber computation to learn to predict reward. For over 50 years, it's known that the cerebellum implements a form of supervised learning to learn to predict an action based on a cue. So in the cerebellum, the input arrives via mossy fibers to the granule cell layer, which extracts relevant information from the input and conveys them to Purkinje cells. Purkinje cells then adaptively adjust the strength of granule cell connections to generate a response. And they do this by, using an instructive signal computed in the inferior olive and conveyed to Purkinje cells via climbing fibers. The output of Purkinje cells is then transmitted to the rest of the brain. We also know that Purkinje cells adjust the strength of granule cell connections using a plasticity rule called long-term depression, where granule cells that are active within 150 milliseconds of a climbing fiber spike are depressed. Okay, so a model of cerebellar learning called the ley lines model proposes that in order to keep track of time between a cue and an action, one has to generate granule cell timing bases. So I'm going to show you an example of a task. This is called eye-blink conditioning. So in eye-blink conditioning, a conditioned stimulus is presented, in this case, a tone, and is followed by an unconditioned stimulus, in this case, a nerve puff to the cornea, which triggers an unconditioned eye-blink response. So after paired presentations of the conditioned and unconditioned stimulus, the eye-blink becomes conditioned so that it precedes the onset of the air puff. And so we know that the cerebellum is responsible for synchronizing the tone to the action of the eye blink. And this can happen as fast as within 100 milliseconds. So this lines model proposes that a non-overlapping sequence of granule cells encode time, and using a climbing fiber error signal, the Purkinje cell output is then generated at the appropriate delay time to trigger the eye blink. However, this model does not account for tracking the passage of time throughout a delay using ramping signals that have been found in the cerebellum and elsewhere in the brain. So this is important because the cerebellum has been implicated in a variety of cognitive tasks that track the passage of time, such as active maintenance. So we set out to investigate how does the granule cell basis encode time and how Purkinje cells integrate this information to generate a ramping signal that tracks time. So we recorded from granule cells and climbing fibers as these two inputs converge onto Purkinje cells. And we did this using calcium imaging. Here you can see a video of the calcium transients recorded simultaneously. Okay, so we did this in head-fixed mice that performed a behavioral task, which consisted of mice pushing a lever, and they obtain a water reward after 1 to 2 seconds. So keep track of this delay because it's gonna come up repeatedly in the talk. So we found that mice learned the timing of the expected reward from their licking behavior. So on day 1 of training, mice licked more following the onset, following the forwarding movement, sorry. After 7 days of training, they learned to shift their peak leaking to the expected reward time 1 second later. So when a reward was unexpectedly omitted, mice stopped leaking after they presumably realized that excessive time had passed and a reward was not likely to be delivered. We also trained mice on a 2-second delay task, and they learned to shift their peak leaking accordingly. Okay, so you're looking at the granule cell activity here. These are 3,500 granule cells that we recorded from 20 mice. On yellow, you can see where granule cells became active during the behavioral task, and you can see that many granule cells became active during the delay. Climbing fibers, on the other hand, became active mostly at the reward delivery time. So this is a subset of the granule cells that were active during the delay, and you can see that activity spans the entirety of the delay and shuts immediately off after the reward was delivered. So for mice that we train on a 1-second task, you can see the timing basis formed by granule cells here that spans the 1-second delay. And for the 2-second delay, the granule cell activity adapted to span the entire 2-second delay. These observations are already different from the delay lines model that I talked to you about at the beginning of the talk, where granule cells are activated in a non-overlapping sequence. So how are granule cells encoding this timing information? As I told you at the beginning of the talk, we know that granule cells that are active within 150 milliseconds of a climbing fiber spike are depressed. So we applied this plasticity rule in our data. So for every climbing fiber that spiked following a reward, we predicted the amount of long-term depression each granule cell underwent throughout an entire training session. So granule cells that were very active during this LTD window in green underwent more LTD than cells that had low activity during this brief window. So we then grouped cells, groups of granule cells, based on the amount of LTD they underwent throughout a session. Where in magenta, we have cells that underwent very low long-term depression, and in light blue, cells that underwent a lot of long-term depression. So you can see this granule cell timing basis that spans the delay, and it's refined through a climbing fiber signal. And so this information, we think, is conveyed to Purkinje cells where they can provide information up to 1 second into the future in expert mice. This timing basis falls through immediately following reward. But for mice that were trained on a 2-second delay, the timing basis adapts to stretch the entire 2-second delay. So how are Purkinje cells integrating this information? We performed a very simple computation where we summed the predicted LTD magnitude of every granule cell, and to predict what the Purkinje cell output would look like, we ended up with a downward ramping signal consistent with our prediction. So this downward ramping signal in 1 second expert mice fell through following reward, but for mice that were trained on a 2 second delay task, the prediction of the Purkinje cell output continued ramping throughout the delay. So finally, we recorded from Purkinje cells while the mice were performing this behavioral task. We did this again using calcium imaging. And we found downward ramps throughout the 1 and 2 second delays, consistent with our prediction. So to conclude, the delay lines model that proposes non-overlapping sequence of granule cells to track the passage of time does not account for tracking time using a ramping signal. We then found a granule cell timing basis that adapts throughout learning to span the delay instructed by a Clavian fiber signal, reward evoked Clavian fiber signal, and long-term depression to result in this ramping output that tracks time. So I would like to thank all of my co-authors, my PI, Mark Wagner, all of our funding agencies, and the Cosyne organizing committee for awarding me with a travel grant. Thank you, and I will take any questions.
All right, 1 or 2 questions before the coffee break.
Hi. Here. Really cool work. The question I had is the granule cell time basis seems to be adapting to the delay. What is the source of this? Is it happening within the cerebellum? Is it an internal process? Or do you think the entire time coding and the creation of the basis is something that's happening from the outside of the cerebellum?
Yeah, that is a great question. We haven't actually tested if it's inherited from other areas in the brain, like the cortex, but we know that the timing information is encoded itself in the granule cell layer. Like, information in the granule cell layer is sufficient to support this computation.
Thank you.
Thank you.
Hi, I just have a question about the behavior. So in the, excuse me, in the example you gave of eye blink conditioning, that's a Pavlovian task, and then you guys picked a Skinnerian task. Do you have a reason to believe that the mechanism of timing would be the same or different between a more unconscious Pavlovian task versus a conscious Skinnerian task?
Yeah, I mean, it would be good to find out what the granule cell basis actually looks like for an eye blink conditioning task. But it might be the case that it's not necessary to track time, so it might be a different granule cell basis.
So why did you pick a Skinnerian task as opposed to a Pavlovian task when we know like the cerebellum is deeply involved in eye blink conditioning?
Well, I mean, we wanted to move to study more cognitive tasks, and so, and also a Purkinje cell output that would support a ramping signal. Yeah.
Thank you.
Is it a short question? Yeah, okay.
Hi, great talk, thanks. So I see in this last figure the sustained activity in the granule cell population or across neurons. I was wondering what gives rise to that sustained activity, because it's relatively a feed-forward structure and there's not much recurrency, so what do you think could lead to that sustained response?
Well, so far we only have the observation that it actually sustain, like granule cells sustain activity. We don't know why, but there's a lot of redundancy in the system, which is contrary to a lot of cerebellar theories. Yeah, I don't have a good answer.
Thanks. All right, let's thank our speakers again. All right, now is the time to get rewarded. We'll reconvene at 11.